So now I'd like to hand the microphone over to Associate Professor Marion Harris. Thanks, Marion.
Thanks, Vanessa. Well, without further introduction, I'll get going. And just some background to start. So genetics is a branch of biology that deals with genes and heredity, which is the passing of traits from parents to offspring. So a gene is a specific sequence of DNA that codes for a specific protein that has a role to play in the cell. And we inherit 2 copies of each gene that we have, one from each parent. And gene changes in particular that predispose to epithelial ovarian cancer can be inherited from the maternal or paternal branch of the family tree. I just wanted to highlight with this slide the difference between something that's genetic and something that's hereditary. So sometimes I hear people, particularly young people, say, the doctor said my cancer was genetic, when what they really meant is hereditary. So every single cancer cell has genetic alterations in its DNA. Only in a smaller percentage of cancers is there an alteration in the DNA of the egg or sperm that is transmitted from one generation to another that predisposes to an increased risk of a cancer type. So all cancers are genetic or have genetic changes, but only in a smaller proportion of cancers can there said to be a hereditary predisposition acting. Just wanted to touch on what is the cause of cancer. In short, we don't know, but a series of alterations or mutations in critical genes that control cell growth and division, or in genes that control repair of errors in DNA replication. So an alteration or change in the code in a gene results in a protein that is somehow altered. It may be absent, it may be shortened, and that protein doesn't work as it should. It doesn't do its job. As a consequence, cancer can develop. And we don't really know why. For example, a BRCA1 gene change causes cancers, particularly at some sites like breast ovary and not at other sites. Just touching again on sporadic versus hereditary cancer. So in most people, cancers are not hereditary. Genetic changes resulting in cancer occur due to random error, due to bad luck, so a DNA copying error. If we had to copy out the pages of a phone book each time, as a cell does to replicate, we would also make errors. So when we make errors, our body has systems to correct them. So most genetic errors are due to random bad luck, although these may be influenced by environmental exposures or lifestyle factors, the simplest one being cigarette smoking, but also for female cancers, hormonal factors in particular. So these are sporadic cancers. We don't know specifically what's caused that gene change that caused that individual breast cancer to arise. These are sporadic cancers. But as I've already touched on, if there's an alteration or error in a cancer predisposition gene, then that gene change can pass down to different family members through generations. This isn't a hereditary cancer, but it's the predisposition to cancer is hereditary. Just touching for background the types of ovarian cancer. So when we speak of ovarian cancer, we're commonly referring to epithelial ovarian cancer, which is said to arise from epithelial cells on the surface of the ovary or fallopian tubes. These epithelial ovarian cancers are the main types associated with hereditary predispositions, and these make up about 90% of ovarian cancers. There are 2 other main types, germ cell tumours that arise from the egg cells of the ovary, which aren't typically hereditary, and sex cord tumours, which arise from the tumours like the connective tissue of the ovarian organ. And those latter 2 types, with a few exceptions, are generally not hereditary. Just a reminder that although I refer to ovarian cancer, it's believed that most epithelial ovarian cancer starts in the fallopian tubes, in the fimbria, at the ends of the fallopian tubes, and the cancer cells migrate along to the ovary, or more rarely arise in the peritoneum. Just a reminder, the high-grade serous epithelial ovarian cancer is the most common type, a rarer type being a low-grade. Now, the difference in cells is how they look, how a pathologist would grade them under the microscope, how disorganised, the higher grade being more disorganised, and the lower grade more mimicking normal epithelial ovarian tissue. Mucinous, where mucin droplets are visible in the cell, a rarer type. Clear cell, big white cells, and endometrioid are less common types. So hereditary conditions associated with a risk of epithelial ovarian cancer, the main focus being BRCA1 and 2 breast ovarian cancer syndrome and Lynch syndrome. These are two highly penetrant conditions that confer an increased risk of ovarian cancer as well as other cancers. Then there's also PALB2, which is a relatively novel gene in the ovarian cancer space that we're routinely offering testing for now in familial cancer clinics, and we know that it's associated with about a 5% lifetime risk of ovarian cancer. Other genes, RAD51C and D, BRIP mutations, and then there's something that I'll just touch on, polygenic, hereditary predisposition to ovarian cancer is something that's been unravelled. And then there still remains some people with a family history of ovarian cancer where the specific basis of it is not presently known. So going first to BRCA1 and 2, these are damage-repaired genes. They act as tumour suppressor genes. By maintaining the genetic order or correcting errors, they help to prevent cancer. The mutations or alterations in these genes, because we all have these genes, some people have an alteration that interferes with the protein produced and hence the function. They're inherited in an autosomal dominant fashion. And this just means that if you only inherit one altered copy, you have an increased risk of developing the condition. This contrasts with some conditions like cystic fibrosis, where an affected person needs to inherit an altered copy from both parents in order to develop the condition. So the idea is these people inherit one altered gene copy, and then a second hit occurs, knocking out their second gene copy, meaning that their damaged DNA isn't repaired, with resultant genetic instability, which is believed to predispose to cancer. And as I mentioned, for reasons not entirely clear of, these are mainly confined to the breast ovary in women, and also the prostate in men, as well as some other things like pancreas and melanoma. So it's thought about 1 in 400 individuals carry a BRCA1 or 2 mutation. In people with Ashkenazi Jewish ancestry, it's thought to be about 1 in 40, which is about 2.5%. There are so-called founder or common mutations in other people of different ancestries like Dutch and Indian. And we think that approximately up to 20% of all epithelial ovarian cancer diagnoses, there's a BRCA1 or 2 gene change. So BRCA1 or 2 mutations explain up to 85% of epithelial ovarian cancer in women who have a family history. So if there's 2 women in a family with ovarian cancer, the odds are that it's a BRCA1 or 2 gene change. So BRCA1s and 2 are both associated with up to a 70% lifetime risk of breast cancer. For ovarian cancer for BRCA1, it's thought to be a 50% lifetime risk. That's classically high-grade serous. And for BRCA2, the risk 10 to 20% lifetime risk. As I said, it's usually high-grade serous, but it can be endometrioid or clear cell. We don't typically associate low-grade serous ovarian cancer, borderline tumours, or mucinous epithelial ovarian cancers with BRCA1 or 2 mutations. So these cancer types are not usually offered BRCA1 or 2 gene testing. Some of the indicators we look for a personal or family history of breast cancer in close relatives, particularly young age or triple negative. We look for other people who've had high-grade serous epithelial ovarian cancer, ancestry, high-grade prostate cancer, as determined by the Gleason score in the family, can be an indicator. Pancreatic cancer in the family. The age of the person, so BRCA mutations are less common in women diagnosed under 30 or over 70, so more typically in that middle range. And as I said, more typically serous, but can also be an endometrioid and clear cell. So all invasive epithelial adenocarcinomas of the ovary that are not mucinous and are not borderline are generally considered eligible for BRCA testing. Some units apply an upper age limit of 65 or 70, but not all. Yolan's going to talk about the therapeutic aspects, how having a mutation affects someone's treatment, but just so touching on the effect for relatives. So where a BRCA gene change is identified, there can be predictive testing for unaffected relatives, giving them the opportunity to undertake preventive actions, whether it's breast cancer screening, breast surgery, and to consider bilateral salpingo-oophorectomy by age 40 for BRCA1 carriers and by 45 for BRCA2. It's thought that a small risk of primary peritoneal cancer remains. Then for men, there can be PSA screening. So some figures, I've said it can be up to 20% risk of having a BRCA mutation. If someone has an epithelial ovarian cancer, depending on the age and the exact type, it can be sort of 13 to 16%, primary peritoneal cancer, say 15%, fallopian tube 20%, a lower chance of a mutation if there's a clear cell or endometrioid cancer. And for people who are over 65, the risk of a mutation being identified is lower. So instead of 15 to 20, it's more like 8%. So just moving on from BRCA1 or 2 to Lynch syndrome, which is a hereditary cancer predisposition that explains about a smaller proportion than BRCA of hereditary ovarian cancer. So this is due to a mutation in one of a number of so-called mismatch repair genes that are also involved in DNA repair and also have autosomal dominant inheritance, meaning you just need to inherit one altered copy to have that predisposition for cancer. So the typical cancers for this condition are more colorectal endometrial cancer, and ovarian cancer is a less frequently observed. There's often a multi-generational history of cancer, but not always. The types of ovarian tumours are different. More typically, these are endometrioid, clear cell. They can be mucinous or borderline. They're less likely to be serous tumours. They can occur but are uncommon. And classically, epithelial ovarian cancer in Lynch syndrome occurs at a younger age. And pathologists can screen for Lynch syndrome in people with ovarian cancer by doing a special stain, although in isolation, it's not routine to always do this. So once again, Yolan's going to touch on the therapy options, but in terms of someone who's found to have these gene changes and for their family members, they may need to consider screening for other cancers like bowel cancer. And also their relatives, if they have the same gene change, similar thing, might need to consider preventive salpingo-oophorectomy or colonoscopies. So just touching base on some of the more rarer things. So I mentioned, so many of these proteins work together to interact with BRCA1 and 2. So PALB2, which could also be called BRCA3, is partner and localizer of BRCA2. So it works in together with BRCA2, hence it causes a similar issue of an increased risk of breast and ovarian cancer. RAD51C and D do the same. There's another gene that's called BRIP, and these are all three, all inherited in the autosomal dominant fashion. The lifetime risk of ovarian cancer is what we call low penetrance, meaning there's only a 5 to 10% risk. In contrast, BRCA1 is said to be a high penetrance, where if you have the gene change, you can have up to a 40% lifetime risk. They all three are associated with some increase in breast cancer risk, and there are preventive options for family members that carry the same gene change to have a salpingo-oophorectomy, albeit at a bit of a later age. Just touching on these, CHEK2 and ATM are the names of another couple of genes that are particularly associated with breast cancer risk. And the exact increase in ovarian cancer risk is unclear. As I mentioned before, there's a group that remain unknown. The genetic basis and mechanism of inheritance is unknown. I'm just going to touch base here on something that's called polygenic risk. So in contrast with BRCA1 or 2, it's what we call a monogenic or a single gene risk. There's the emerging ideas for a number of cancer types, particularly prostate and breast. The idea of these SNPs, which are single nucleotide polymorphism, or sites in DNA where variation is common. So we think of inheriting these relevant SNPs like a hand of cards. and each individual SNP increases or decreases the risk of a disease, which in this case we'd be talking about ovarian cancer, but these have also been studied in prostate cancer or cardiovascular disease. Each SNP increases or decreases your risk of disease by a small degree, but it's the combined effect, it's like a hand of cards, of the individual's multiple relevant SNPs relevant for ovarian cancer that determine their polygenic risk for that outcome. So some people will inherit a low-risk combination, a low-risk hand of cards, and some will inherit a high-risk combination. Most people will inherit a medium risk. So this is increasingly being understood and studied in a research setting and may be part of our genetic testing for people with ovarian cancer in the near future. Yolan's going to touch on this, so I'm just going to introduce it. So when we talk about germline genetic testing, we're talking about blood or skin testing, and it's the genetic makeup of those cells are thought to be representative of all the cells in the body, which will include the egg cells in the ovary or the sperm cells in the testis. In contrast, somatic genetic testing is testing of tumour tissue. And while typically therapies are for germline BRCA mutations, testing and finding somatic or tumour alone BRCA mutations can also enable access to therapies like PARP. And just a reminder, so a mutation is an alteration or a change in the DNA. Pathogenic is disease-causing, a disease-causing alteration, and a variant of uncertain significance means it's uncertain if the genetic change has any impact on the functional health of the gene product or protein. So we all carry alterations in our DNA, and many of them are not significant, but some of them are. And we all carry a lot of variants, and it is quite a process and a science trying to work out if these genetic changes are pathogenic or if they are variants of uncertain or indeed of no significance. I just wanted to say this is a typical ovarian cancer panel that we would request pretty much throughout Victoria for most people with ovarian cancer. So as you can see, it's currently a 12-gene panel, including BRCA1 and 2. These 5 all relate to testing the different genes involved in causing Lynch syndrome. P53 is thrown in. It's more typically a breast sarcoma, but can sometimes cause cases of ovarian cancer. PALB2, I mentioned a 5 to 10% risk, same with BRIP and RAD51C and G. So this is pretty much a standard panel. And this is my last slide, but just going through, this is just one series from a few years ago that reported over 4,000 women who had a germline test, and they were tested for 32 genes. And this is just an example where 13% of the group had a disease-causing variant or pathogenic mutation detected, and that was a bit low. They were perhaps thinking more, might have been 16 or 20%. 25% had one of these variants of uncertain significance, and these generally need to be followed up. And over a period of time, some of these will be downgraded to be just variants or nothing significant. Others of them will be upgraded and determined that it's actually now disease-causing. And over half have a negative or what we call an uninformative result. Out of this group, about 20% had a family history of ovarian cancer and about 13% had a prior history of breast cancer. Of the mutations found, as we said, the biggest player, BRCA1 and 2, 54%. A little bit surprising for me, so this CHEK2 that I mentioned, 10%. Then the mismatch repair genes causing Lynch syndrome at 9%. And then the other genes that you can see, so that ATM, more typically a moderate breast cancer gene, BRIP for ovary, RAD51C and D making up a few percent. So look, with that, I'll conclude there and hand back to Vanessa. Thank you.
Thank you very much, Marion, for that extremely informative presentation. We just have a couple of questions for you. One is, if someone does have ovarian cancer and they have not undergone genetic testing, how do they go about getting it?
So it would probably be good to touch base with their treating doctor to just clarify, have they had genetic testing? But generally they need to sign a specific consent form. So if the person doesn't recall doing that, they probably haven't had it. They can always ring, so testing with their clinician, but they can also always ring a familial cancer clinic in whichever state of Australia they are to make an inquiry, and But generally they would need a referral from their GP to discuss having genetic testing if they've not already had it. So Googling Familial Cancer Clinic in their state would be the way to start and getting a referral.
And what is the cost involved?
So generally there's a Medicare item number, so there are a few restrictions, but generally most people with epithelial ovarian cancer, particularly high grade, are automatically eligible for genetic testing so that it is covered. It's basically free, covered by their Medicare cover, so there's no actual charge. If someone wants to have testing outside guidelines, for example, if they had a mucinous ovarian cancer or if they were unaffected but had a family history, they would have to pay for it. And so depending on the panel, it might be up to about $500, but that is a lot cheaper than it used to be. With all breakthroughs in genetic testing, can be done much faster and more cheaply. So in the past it would have been a few thousand, now it might be about $400 or $500. But for most people it is free.
Great, that's great to hear. Thanks, Marion. There's just one more question. Is HRD testing going to be available in Australia soon? It wasn't an option when I originally had my testing done. This is from one of the participants.
So at the minute, that's very topical. So I understand that there was sort of a patent held by a company in the States. So I know some people were paying for their samples, tumour samples and blood samples to go over to the States to get a report back to then determine, whether they should look at, self-funding, one of the PARP inhibitors. I believe there are moves afoot to make a different type of HRD testing available in Australia. So at my own centre, I believe deals are being done to enable something to be available in Australia. But I suppose if someone gets an HRD test, you sort of need a therapy that's available. So at the minute, the Australian guidelines say you need to have a BRCA mutation. So if you have an HRD test that's positive, but you don't have a clear germline or somatic mutation, it might be tricky to get a Medicare-funded subsidised therapy, but of course people can fund it themselves. So look, in short, I hope that it might be around the corner. I don't think it's going to be in the next month, but I would certainly hope in the next 6 to 12 months. So watch this space. And I don't know if Yolande had any particular comment to add there.
We might move on to Yolande, so she can probably answer that